Thanks everybody for coming, and thanks Anna for the nice introduction. Welcome to the first Fluorescence Neuro-imaging and Photonics series of seminar. And Anna has mentioned before, remember that you can ask questions in the chat at any time, and they will be read and answered at the end of each talk. We are very lucky today to have an exceptional lineup of speakers who will tell us about the latest developments in methods for imaging processing. So the first speaker of today will be Professor Andrea Giovannucci, and now Marco will introduce him. Thanks again for being here today.
Hi everyone. So today we are delighted to have here Professor Giovannucci. Andrea, after his PhD at the Artificial Intelligence Research Institute of Barcelona, joined the Princeton University and Pompeu Fabra University, where he worked on computational neuroscience, neuroprosthetics, and experimental neuroscience. Then he took on a data scientist position at the Flatiron Institute, the Siemens Foundation, to develop machine learning algorithms and open source software for the analysis of calcium imaging data. He is currently assistant professor in neuroengineering at the UNC and CSU Department of Bioengineering, the Closed Loop Engineering for Advanced Rehabilitation Center, and the UNC Neuroscience Center. His current research interests focus on the analysis of biomedical neuroscience imaging data, including calcium and voltage imaging at cellular level. Today we'll discuss with us about an accelerated pipeline for fluorescence imaging and online analysis he recently developed. So without further ado, Andrea, please, the virtual stage is yours.
Thank you for the kind introduction. Thank you to the organizers for this fantastic event, this series of events. And I feel very privileged and honored to be invited to share my research here with you today. So can everybody see nicely my screen? Cool. Okay. So as you know, as we mentioned today, I'm going to focus mostly on real-time analysis pipeline for calcium and voltage imaging data. So this is the outline of my presentation. I will give you a very, very quick primer on the analysis of fluorescence imaging. What are the issues that are related to the preprocessing step to go from raw frames to spikes. Then I'm going to introduce very quickly Cayman Online, at least a small aspect of that, and that will bring me to discuss what is the current research of my laboratory, which is FIOLA. Okay, I don't think a lot of introduction is required here to motivate why calcium or voltage imaging are very important. But briefly, calcium and voltage imaging are techniques that are relatively young, especially voltage imaging. But calcium imaging basically enables you to track large neuronal populations across weeks with single cell resolution, and also targeting genetically subpopulation of interest. We can do these experiments on behaving mice and other preparations. And the idea normally is that you can image both somas or compartments of the neurons, and what you need to do is to go from these basically little donuts or dendrites in this case, to the activity of these neurons. So the transformation from one to the other is nonlinear and requires some non-trivial image processing steps. But the idea is basically to go from this little donut turning on to spikes. Voltage imaging, here I have an example. Recently, indicators have improved, and now we have access to, tens of neurons at the same time. In this case, with millisecond time resolution. This is an example from a paper we recently published. But what you see now is that you can again gain access not only to spikes but also to signals that are subthreshold. So voltage is also a very promising modality that is growing a lot recently. The same can be said about voltage imaging in terms of preprocessing steps. You need to go from something that might look like this or this to spikes, right? And eventually subthreshold signals. There are two ways in which one can analyze data. One is to have the full movie available at analysis time. You can access it and analyze everything from beginning to end. which is what you normally do, when you go to sleep by night after a long experiment, right? You send it to some cluster or to your computer, you let it run, which is amazing. But what we are also interested in a new type of modality analysis, which is frame by frame. Okay, what does it mean? What does it mean? Instead of having available the whole movie, either in memory or materially not available the whole movie at some time point, what you can do is process it in streaming. That is frame by frame, right? We call this modality online. The modality of the whole movie, we call it patch. But why would you have access to a single frame at each time? Well, one example, for instance, let's put ourselves in the situation of, for instance, 2-photon imaging, is that you might want to do closed loop experiments with optogenetics, right? So you want to know what the brain state is at any given moment, and then modulate the activity of neurons. Not only, you might even want to use this to calibrate your optogenetic stimulation and provide a function from stimulation to elicited spikes or elicited fluorescence. Other interesting experiments are, of course, controlling neuroprosthesis, in the future control computers as well. So, of course, this is on the long term, but the idea of having access to the activity at a specific time point has multiple applications. Now, the requirement to extract in real time this data are tight, right? What you need to do, you have to correct for motion, right? So this means that the brain is moving under the microscope. And what you want to achieve is a situation in which each pixel will always represent the same spatial location. Because if this doesn't happen, let's say that you take an average of this neuron here, as this brain moves under the microscope and you're capturing the signal, you will have a lot of signal that is unrelated to the activity of neurons, but just to the movement of the brain. Afterwards, you need to localize neurons. So you need to be able to have an approximation of where neurons are and what is the spatial extent. Once you have that, you want to extract the fluorescence from these neurons. Now, you might think that this is trivially a segmentation problem, which we segment this neuron, we segment this other neuron that you see in yellow. When we take the average of one and take the average of the other. Now you can clearly see from this movie, when you do this approximation, you take the average of one and the average of the other one for the activity, you will commit a big mistake, right? Because the activity, the intersection of the two neurons will contaminate the activity of each other. So what you will have is that you need to solve, it's called a source separation problem, that is assigning to a set of pixels, right, the activity of each neurons, paying attention not to contaminate. On the top of it, if you look at the background here, you'll see that also there is a fluorescent signal associated to it. If you think again, in this situation, pixels in the center here will have a mix of activity from the red neurons, the yellow neurons, and the background. Finally, once we have demixed the activity, we need to extract the spikes or an approximation of the spikes, which we call a deconvolution problem. Okay, so what is the framework? Let me focus today mostly on the problem of separating sources and identifying neuron location, okay, for in the interest of time. So what is the model? What is the computational model associated to this demixing problem? Well, the idea is simple. You want to represent a movie as the sum of multiple components. Components will have two types. One is going to be the spatial components, which represent the spatial footprint associated to each of the neural entity, and the temporal component, C, which represent the fluorescence. There will be another class of components which will represent the background signal, the neuropil activity, okay? With the same characteristics, a spatial component and temporal component associated to it. The computational framework is called constrained non-negative matrix factorization, which simply means that you are trying to approximate this matrix as the sum of these two matrices with some constraints. For instance, sparsity on A. What does it mean that it's sparse? The spatial footprint are sparse because they're very localized. You just find them in a small portion of the field of view. Non-negativity. Non-negativity stems from the physical meaning of photons that there are some and cannot be subtracted from each other. Yeah, let's say a practical example. If you think about the movie here, what we do, we vectorize the pixels. That means we start from 60 by 80, and we actually have the whole number of pixels times the total number of frames. This is a big matrix. And what we try to do is to approximate it by the multiplication of thinner matrices. And this, when we solve this optimization problem, naturally, actually what we obtain is a demixing of the properties, of a demixing of the neuron associated to each of the components. For instance, we take one column from this matrix, which are the spatial components, we reshape it, the shape of the frame, and it looks exactly like a neuron. You look at the temporal components, you see that it extracts a very clean temporal component. And you can understand what I told you at the beginning, that the sum of many components, like neurons, are represented in this movie, because when we perform the outer product between this column and this row, what we obtain is actually the contribution of that single neuron to the whole movie. So now imagine you have a set of neurons, each of them will contribute to the movie. They will be demixed by the solution of this optimization algorithm. And then at the end, you will also fit in other components which represent the background. Okay, it has exactly the same meaning. You know, if you take this column, you reshape it, it will look like the background, the neural pill activity, spatial footprint, and the neural pill activity temporal profile. Example of a movie that is analyzed completely. You will have a set of spatial components, a set of temporal components. On the top left, you have the raw movie, the original movie. And on the bottom right, you have the movie that is reconstructed by adding only the neurons. If you remember my previous slide, I told you that you can actually represent each neuron with this outer product, and now you sum all of them, and you obtain a very clean version of your movie in which only neurons are visible. If you also add back also the background too, then we obtain is a denoised version of the original movie. So it will contain most of the information that is contained in the original movie, but denoised. Now, if you take the difference between the raw movie and this reconstructed movie, we get the residual, which is here at the bottom. And if we did a good job, if we were able to find everything that was in the field of view, the residual should look like mostly white noise. Okay? And indeed, it seems that you're doing a reasonable job in this case. There are still some residual activity, but we were able to capture most of that. And now notice, we're using much less elements than before, because the matrix of the original movie is very fat, but what we have is just the product of much thinner matrices. Why did I tell you all this? Because I want to tell you, how do we solve this problem in real time? Because now we want to be able to solve the problem of correct information in real time. We want to be able to separate the activity from neurons, extract spikes in real time. However, when we are dealing with real-time scenarios or online experiments, other requirements emerge. For instance, in the hippocampus, some neurons become active maybe half an hour or 1 hour after you started the experiment. So you want to be able to incorporate the neurons that were not previously active. Similarly, on long experiments, neurons might slightly change shape because of hydration of the animal, for instance. So you want to be able to adapt the shapes of the neurons, so it's always optimal with respect to what you see in the field of view. Not only that, you also want to be sure that you can accumulate information about the activity of neurons. So let's say if the neurons becomes active at times t just once, you know, if it becomes active again at times t plus 2000, you want to be able to accumulate the information to clean up your spatial component. So you want to be able to refine your spatial component given the activity of neurons at multiple times, at multiple positions in times. Now, this, you know, there is no existing methods that can address this problem online. This is the only work that address this gap, and also does so in a big data regime with data streams ranging from 30 to 110 megabytes per second. Okay, so what do we do when you do a real-time experiment? So the usual pipeline is initializing with, let's say, 1,000, 3,000 frames, using the algorithm that I mentioned to you at the beginning, the constraint non-negative matrix factorization. Then you motion correct frame by frame. In the interest of time today, I'm not focusing on this aspect, but you are welcome to check this paper from Journal Neuroscience Method 2017 if you want more details. Today we're focusing on the portion of estimating spikes, more appropriately said, the source separation more than the spike estimation, detecting new neurons, the appearance of new neurons, and how shapes are adapted. You can find this information in detail in these two papers that are mentioned at the bottom. Okay, let's start with spike estimation. We are in the situation in which we initialized our algorithms with, let's say, 1,000 frames. We found a set of components, spatial components that you find on the left, a set of temporal components. Now we are here at the beginning of the shaded area. And we start our experiment. So what do we need to do? Well, we need to fit, for each time point, the activity from each frames to each of these spatial components. Now, what does it mean? We try to explain the activity. I'm going to back again to the metaphor of explaining the activity. We try to explain the activity within the frame, provided the neurons they already found. So if I do this, in the residual, if you look at the residuals, that is the difference between what I estimate and the original frame, I should see nothing or noise. So that's the assumption. Now, this problem is very easy to solve. It's not very easy to solve, but we found a very efficient way of solving this problem. Is a non-negative least square, if you're interested in the details of the algorithm. And we solve it with a block coordinate descent approach with warm restart. I'm happy to discuss this offline if you're interested. Okay, so how do we detect new neuron? And now, this is exactly where it comes to our help, the residual. Why? And again, I stress, if I did a good job to estimate neurons with to estimate the frame with the neurons that I had available, I should see nothing in the residual. Now, what happens if a new neuron appears? In a new neuron is basically something I cannot explain in the frame. And indeed, since I cannot explain it in the frame, and I look at the residual, in this case, the average of many frames of the residual, what I will see is something that looks like a neuron emerging out of the blackness. That's exactly what happens here if you look at this green arrow. So what do? We develop the methods that scans the field of view for promising regions. Then we train the convolutional networks, simple convolutional networks, to identify neurons. Basically, squares that will look like somas versus squares that will look like either noise or processes, or let's say something we are not interested in the analysis. Of course, this can be extended with arbitrary, type of, let's say, anatomical features, provided training data, of course. Finally, if we do this, we reach our goal, the stated goal, which was, if a new neurons become active, I want to be able to capture this activity. And you will see it, you see down at the bottom there, in the bottom right square, you see that when the neurons in the green arrow becomes active, we are able to capture it. So we can use it in a closed loop setting, for instance. Finally, we need to update shapes. Okay, so if you look here, the simulated data, once we find a new neuron at the beginning, we have not much information to describe the shape. So what we want to do is to accumulate information, and that's exactly what happens in this algorithm. On the left, you have an example on real data, which the neurons that is indicated by the red arrow become active. And shortly after the neurons in the red arrow become active, you see at the bottom that the spatial component is updated. So this is done. So now the computational steps required to perform this operation are actually very technical. What we use is a framework that is called online dictionary learning. And again, if you're interested, I can get into the details afterwards. So summarizing, what do we do with an online experiments? We initialized on about 1,000 to 3,000 frames that we start our experiments. We correct for motion each frames. I didn't get into the details, but you're free to ask me or check the paper. Then we need to separate the activity from overlapping neurons. And we do so by solving a non-negative least square problem. Finally, we need to identify new neurons appearing in the field of view. And we do so by, let's say, employing computational vision tools on the residuals. And finally, we want to update the shapes to be sure that we're always tracking, a clean version of the spatial footprint. And you can obtain at each step the activity of each neuron, which was our originally stated goal. It's an example of real data. This is mesoscopic data. On the top left, you see the raw data. Here you see the residual and the convolutional neural network that finds neurons. We are accelerating, of course, this artificially. And then, here you have just the activity of neurons or the denoise data. An example that might be dear in this audience is the zebrafish. Basically, here is 40 planes from an experiment with zebrafish on a light sheet. We sent each of these frames to a node in a cluster, and we were able to analyze the full data. This is about 400 gigabytes in just 15 minutes. So basically, we were able to analyze it at 2 Hertz, and it was acquired at 1 Hertz. In principle, one could also do closed-loop experiments with a zebrafish, with the caveat that we are analyzing each plane separately and not in 3D. Okay, so when you look at the performance of these algorithms, I think there is a question. Okay, maybe I can answer this one later.
Yes, we can address the question after your talk.
Beautiful. That's a good question. Very good question, by the way, already. I see that. Okay, so so what we do is we have time per frame in a standard, if you want, setting, which is a resonance scanner running at 30 Hertz. There are about 200 neurons in the field of view. What we see that, we are doing well at the beginning. Then as we find more neurons, the timing gets worse. Right? And then, 30 milliseconds is the limit. And, a few neurons really are getting a bit over the 30 milliseconds. So, it can be used in online scenarios, but with the caveat that computationally, it's still pretty expensive. And this brings me to what my laboratory is doing now. Not only because we want to accelerate, also there are new indicators that are being produced that will enable decoding spikes at much faster speeds. So for instance, the new XCamps, but also GCamp8F, they allow you to decode spikes that are 10 to 20 milliseconds apart, which is different from what we have now, that is at least 50, 60 milliseconds apart. So we can increase the imaging speed to another level. Not only that, if you think about voltage imaging, even more so. Voltage imaging, you can now enable spikes that are decode spikes that are 2 milliseconds apart, leading to imaging speeds that are above 1 kilohertz. So what we had actually is not enough. This is Kaiman working with, let's say, 256, 512, 512, and 76. And so as you add more neuron, it becomes more computationally costly, and there is a big, variability in the times for each frame. So this actually leads to problems in the real-time scenario. We identify the main bottleneck in the motion correction and source separation. So very quickly, I'm going to tell you what we are doing about that and the results that we obtained. What we did, we tried to exploit new tools coming with the machine learning. Machine learning is becoming pervasive, of course, but also tools that are used by machine learning are becoming better and better, like GPUs, right? Or frameworks that optimize computation on GPUs, like TensorFlow or PyTorch. So what we did, we say, okay, let's put this thing together. Let's try to implement our algorithms as they were kind of layers in a neural network, and then let both have optimization on GPU, but also have the computational graphs associated to these operations, so basically the parallelization on the GPU, be taken care by these super advanced frameworks, in this case, TensorFlow. So we adapted our algorithms, both for calcium and voltage imaging, to run on the GPU. So basically, you put the frame at the input in your GPU, and what you get out are the traces. We can then use these traces to do the convolution in calcium, to extract subthreshold activity and spikes in voltage, and so on, depending on our needs. So what do we do in terms of timing? Actually, we are able to boost the timing by a factor of 10 at the very least. But these gains improve as we add more neurons. So for instance, in this case, to process 500 neurons, sorry, I think we swatched the color, sorry. This is 512 by 512. So 512 by 512, 500 neurons, it goes about 300 Hertz now versus the 30 Hertz we had before. And then we check, are we doing well? Because we had to do let's say, to cut some corners. There's some compromises to be taken. For instance, we cannot update the shape anymore. We cannot find new neurons. Are we still doing well with respect to Kaiman online? And the answer is yes. Most of the time, like in this case, these data sets, what is the difference between these data sets and these other ones? These data sets is densely labeled, okay? And there was much contamination. So the more contamination you have in your data, and of course, the more this online approximation becomes less efficient. Okay, let me summarize what we've been seeing so far. I told you a little bit about calcium and voltage imaging analysis. I explained to you there are two, let's say, type of algorithms you can use. One is Kaiman, which will not only extract activity, but also find new neurons and update the shapes, and a new GPU-boosted framework that is called FIOLA, which will analyze data very, very quickly, but will not update shapes and find new neurons. Both of these tools can be used depending on your need. They will enable closed-loop experiments, but also, and I think this is a fundamental use for the future, accelerate the experiment analysis cycle. So now you think you're doing an experiment, it would be great if you could have access in real-time to what's happening there. If you remember, old school electrophysiologists, they were listening to spikes. We are losing this with calcium imaging. So it would be nice to recover that dimension of immediacy during the experiments. And finally, we are integrating this with holography to perform closed-loop experiments. I would like to thank a myriad of people that were key during the development of both Kaiman and FIOLA, especially Eftichios and Johannes, which worked together at the Thymus Foundation in the development of Kaiman, and Chanjia and Cynthia, two exceptional students, which led the development of FIOLA. You can consult both the GitHub repository or papers if you're interested in knowing more about these algorithms. And also I would like to thank my funding sources. And again, you for the attention and for the opportunity. Thank you. I'm ready to take questions.
Well, thank you, Andrea, for the amazing talk and the beautiful movies. And congratulations on the acronym you choose for your approach. So perhaps now we can address some questions. I saw there is already one. Perhaps you can read it or just answer.
Yes. Yes, that's true. So Federico, two things. So first, for the online aspect, of course, we are violating one important constraint, that is the locality. We're scanning the field of view for localized neurons. So definitely this doesn't work well for dendrites. In terms of Kaiman batch results, we used it to segment some type of dendritic signals like Purkinje cells. But I have to be honest, I don't think that's a solved problem in the general case. So I see, for instance, data from apical dendrites in layer 1. That's very difficult to segment for any tools we have tried. But there are algorithms that probably a little bit better than Kaiman are doing this. We used it, but the short answer is that I don't think in the general case this is a solved problem.
Okay, perhaps I have another question.
Yeah.
Do you think there is room to a further development of your approach, including the update of the source separation and the segmentation? What do you have in mind so far? I mean,
are you referring to GPU or just in general?
Yeah, I'm talking about the GPU.
Yeah, there is. We are thinking about that. The technicalities are that when you use TensorFlow, you need to compile a model, and compiling the model has some constraint that sometimes are a little bit hidden to developers. So it's a lot of trial and error. But yes, we are thinking about both things. Updating the components. Actually, what is very natural in this case is finding new components. Because if you think about it, you just need to compute the residual, and you directly can pass the residual to a convolutional network like Mascar CNN to localize promising sources. So that is very easy. The updating of the shapes, that's a little bit more challenging, I think. But we are thinking about that. I think a very interesting direction of development here is volumetric imaging. Because in our experience, that's what really is making a lot of people suffer. These data sets are 1 terabyte per hour or more. They're in 3D, so analyzing in 3D is, you're going cubic now. So a lot of issues with that. So we're also working on making volumetric accessible to GPU, which I think is really is going to boost a lot of groups that are having difficulties right now.
Okay, thanks again a lot. I think it's time for the second speaker. So we're very pleased to have here today Dr. Daniel Sage.